again, that drug, that is about the valdecoxib, can undergo, I don't know whether I had its metabolism in here. I don't think I did. I have celecoxib. But the most important is to note that because of the fact that it has the methyl group can undergo the same metabolism as we have talked about. And then, of course, the sulfonyl group can undergo oxidation. And then you can also have the oxygen and nitrogen glucuronide, because I believe that is because you do have, you see, the valdecoxib has the isoxazoline ring, which has nitrogen and oxygen. So those the nitrogen and oxygen within the isoxazoline ring can give us N and O glucuronides, and which were found to be the major metabolites. Because once you get N and O glucuronides, the glucuronides highly water soluble, you remove them from the body, even before you do the other metabolism. But of course, we have also mentioned what else can happen. Valdecoxib, it may cause bronchoconstriction or anaphylaxis in aspirin-sensitive patients with asthma who have experienced several bronchoconstrictions. So we are only saying it can cause bronchoconstriction. So that means you must be very careful to which patients you give valdecoxib. Very, very careful. You cannot give them to patients who are asthmatic or who are susceptible to aspirin, because they might still have also same susceptibility. Valdecoxib, this is a prodrug. Again, this is a drug which is a prodrug. It's an amide of valdecoxib. It is administered IM or IV for perioperative analgesic inflammatory use. It's a prodrug. It undergoes rapid in vivo hydrolysis to valdecoxib. It's the only parenterally administered coxib. So that is, since we have already done valdecoxib, parecoxib will be metabolized into valdecoxib, and then the metabolism, the complications, the side effects will actually be the same. Etoricoxib, that I hope you have drawn. Do we have the structure of etoricoxib here? Let me get it for you. Etoricoxib. We had all their structures somewhere. Here we have their structures. So if you look at etoricoxib, here is etoricoxib. Again, etoricoxib is also a methyl sulfone. So we have notably two methyl sulfone, etoricoxib, and which other one? And rofecoxib. Two methyl sulfones, etoricoxibs, and rofecoxib. Those are the methyl sulfones. So if you look at this, as I said, it is just another just another demonstration or another illustration that you can have also a 6-membered ring, which is a pyridine ring. The others were 5-membered ring, 5, 5, 5. For etoricoxib, is a 6-membered pyridine ring with a halogen, and I think we had mentioned that for 6-membered ring, a halogen is responsible for maximum activity. So that is, I just wanted us to appreciate the difference between etoricoxib and the others, and the major difference is that etoricoxib has a 6-membered heterocyclic ring, the same way attached at adjacent positions to phenyl rings. And you can see the phenyl rings, even of course this second phenyl ring is not it's also not a phenyl ring, it's a pyridine ring, so it has been substituted, but still with good activity. So it's important to note those structural differences. So we are saying that the structure of these drugs can the structure can be highly manipulated with still giving us important activity. So it is not really the type of ring, it is the arrangement of rings. As we have seen, what we have seen is not changing, is the position at which those rings have been joined. So we are looking at etoricoxib, and we are saying it has a methyl sulfonyl group common to the other coxib inhibitors. That is rofecoxib. Etoricoxib is rapidly absorbed with good bioavailability. Food decreases. Etoricoxib is metabolized, or metabolism involve oxidation again of the 6' methyl group. Again, if you look at that structure, we have seen it has a 6' methyl group, primarily by, again, the cytochrome P450 enzyme. Sometimes you might not remember the specific isoform, so we can generally just say by the cytochrome P450 enzyme. Other metabolites include the 1' N-oxide and glucuronide. It's primarily excreted as metabolites in the urine. So again, the metabolism, as we had mentioned earlier, is usually structure dependent on the structure that you have. Again, then the last one is a lumiracoxib, and you can see all of them, as I have said, all of them are coxibs, because although they might slightly differ with their structure, they do have the same mode of action, and they do have, of course, some structural similarities. You can make sure that you draw the structures of all those. You remember I did say lumiracoxib does carry a slight structural difference. It does have a slight structural difference, and actually very slightly is most has structural similarity with the phenyl acetic acid derivatives like the diclofenac. It has very similar structures, because remember, this is the acetic acid, and then in the other position, it has the amino group. The major difference is that, remember, we had said that for those other drugs like phenyl acetic acid derivatives, the first ring, the ring carrying the carboxylic group is not substituted. So that is the main difference here, that in lumiracoxib, although it has a structural resemblance with the acetic acid derivatives, it is not completely the same, because it's a phenyl acetic acid ring has indeed been substituted. In fact, we consider this is position 1, position 1, 2, 3, 4, 5. So in fact, you can say this is that's also meta. At its meta position, it has been substituted at its meta position, unlike all the others. And that is what is making the difference. And this substitution here, mainly for this, because the mode of action is different, this substitution here by the CH2COH, this substitution is what is giving CH2COH here is for purposes of giving us the rigid group rather than giving us the acidity of this drug. The importance of this group is acting or has replaced the methyl sulfone or the sulfamoyl moiety, and its substitution on that position now gives it the bulkiness, because remember we did talk about bulkiness. Further substitution there gives it the bulkiness it requires to be able to go to enter the COX-2 and not the COX-1. Otherwise, the substitution on the other one is just as we have seen. Of course, we didn't get a fluoro substituent, mainly we got a chloro and chloro. So again, that gives a slight difference. But we can see some structural resemblance. But although there is structural resemblance, let us do understand that the various groups here are serving the correct structure, or the correct bulky, rigid structure, which is required for binding without binding the COX-1 receptor. This we have done now. I should have carried those structures, but I didn't carry them this time. And I'm sorry. I should have carried them as I discussed each drug. So we are saying this is extensively metabolized, reduced incidence of GIT to bring in reduced incidence of GIT irritation, of course, also the kidney challenges. It will have lesser of that. But as we have said, the major challenge that has been identified with the coxib is, of course, the cardiovascular challenges. And we have said this is because they will now block the prostacyclin synthesis more than the thromboxane. The COX-1 mediates the production of prostaglandins and the platelet aggregate stimulant, the thromboxane. COX-2 mediates the production of the platelet aggregate inhibitor, prostacyclin. The adverse CVS effects of the selective COX-2 inhibitors is the consequence of inhibition of COX-2, which results in significant reduction in the production of the prostacyclins, whereas the production of thromboxane by COX-1 is unaffected. So you get higher levels of the thromboxanes and lower levels of the prostacyclins. So there is no balance So when you use like aspirin, you get higher levels of lower levels of thromboxane. When you use these coxibs, you get higher levels of the lower levels of the prostacyclins, and therefore they have been responsible. These drugs have brought about the undesirable effect of cardiovascular disorders, and it can be easily explained by that. Thus, the design of highly selective inhibitors of COX-2 would not be as desirable therapeutically as drugs that preferentially inhibit COX-2, but also inhibit COX-1 to a lesser extent. So we are only saying the better drug is like meloxicam. Better drugs will be like meloxicam, where they do not completely inhibit COX-2, but they do not completely inhibit COX-2, but they preferentially inhibit COX-2, but also inhibit COX-1 to a slight extent. And in inhibiting COX-1 to a slight extent, it means they will also inhibit the thromboxane. They will also inhibit the thromboxane synthase, and in doing so, there will be an balance between the thromboxane and the prostacyclins, and you will avoid most of those toxic effects. So that is our conclusion, that also, yes, the celecoxib are good. The coxibs are good, but even when you go out there, you must be careful that, or even when you dispense them, you must be careful, because we need to be careful that the patient does not have any heart conditions. The patient does not have any heart conditions, because if the patient is already compromised, then you can't bring in a lot of, the drugs can also bring fatality because of the further further complication or further toxicity or cardiovascular toxicity. And I think that in that now we end. Unless if there is a question, I think that gives us the end of the coxibs. And the coxibs are the main selective COX-2 inhibitors. Unless if there is a question, I can move on to the last part, because we've taken twice. On to the last part of the non-steroidal anti-inflammatories. Of course, the last part is the anilides or the paraaminophenols. They're not really non-steroid, they're not really anti-inflammatories, but they have good work, a good activity as antipyretics and anti-analgesics. But their anti-inflammatory activity is very little or minimal or not there at all. So if there is no question, then we move on to the last category of our drugs. We have to put these drugs here so that we do study all the analgesics, the normal analgesics and antipyretics, although these drugs, as I have said, have minimal or no anti-inflammatory activity. When we look at the paraaminophenols, this is where our very, very popularly known drug paracetamol belongs. And paracetamol is a drug we also call acetaminophen. You can see there is Panadol there is a brand name. So when we look at those drugs, we are calling them paraaminophenols. They're also known as paraaminophenols or anilides. And we start with the basic drug, which is acetanilide. If you look at acetanilide is just the phenyl ring plus an amino group there, which is acetylated. That is acetanilide. But acetanilide was to have a lot of toxicity. So indeed, acetanilide cannot be used as a drug, because remember, good activity must be balanced with the bad activity, whereby the bad activity is the toxicity. So acetanilide has a lot of toxicity. This is mainly because this can be easily hydrolyzed to give us aniline. Aniline is known to cause a lot of toxicity in the body, because it will bring about a condition whereby there is formation of Fe3+, and Fe3+ within the hemoglobins does not carry phenacetin. So phenacetin you can't see has the same structure as acetanilide, but somehow it was found that if you add a parahydroxy group on acetanilide, there was a slight decrease in toxicity. And then finally, the same, you see acetaminophen or paracetamol is actually the hydroxyl derivative or the phenyl derivative of or the hydroxyl derivative of acetanilide. And this again was found to have much lower toxicity and also to have very good activity. As we have said, the activity here is mainly analgesic, analgesic, and it's analgesic and antipyretic activity. It is useful for pain and fever, close the door, but not inflammation. As we have said, they have an aromatic ring, but do not have an acidic group, ionizable at physiological pH. So that automatically tells essential, very essential structure activity relationship or structural requirement of having an acidic site. Look at that. Those structures do not have an acidic site. Therefore, they are not able to bind. They are not able to bring about anti-inflammatory activity. They have an aromatic ring, but do not have an acidic group. Ionizable pH, as they do not comply with the SAR, possibly act by some other mechanism. Because remember, we did mention that for these drugs to bind the COX-1 or COX-2, they require an acidic area, but they don't have. So the first drug, acetanilide, is out of market due to toxicity, both blood and liver disorders. That's why I said it is quite effective, but it is quite active, but high toxicity. Then from there, we got phenacetin, which if you look at phenacetin, it has a methoxy group. A methoxy group. It has a methoxy group at the para position. So that was the second group. Was used for decades, but in the 1970s, it was implicated in cases again of liver toxicity and nephrotoxicity, and again was removed from the market. The next then was acetaminophen, is a metabolite of both phenacetin and acetanilide. It's actually a metabolite because if you look at your phenacetin, it has an ethoxy group. If that is oxidative or dealkylation, it will give you a hydroxy group at that position. If your acetanilide undergoes oxidative oxidative parahydroxylation, it will still give you acetaminophen or paracetamol. This was found to be a safe drug, producing much better tolerance and a lower incidence of gastric bleeding compared to many of the other NSAIDs. Difference in mechanism, and of course, definitely because it does not have that carboxyl group, it does not have that acidic group, and as we had said earlier, the toxicity caused by some of these drugs is basically because they're acidic, and therefore they will decrease the pH of the GIT. Structure activity relationship. When we compare acetanilide and acetaminophen, aminophenols are less toxic than the corresponding aniline derivatives. So when they are compared, you find that the parahydroxylation of acetanilide to give us acetanilide of aniline to give us the paracetamols or the acetaminophen resulted in a decrease in toxicity. Paraminophenol itself is highly toxic, so it is not used as a better drug. Paraminophenol is where it's acetamide group. The acetamide group, this group here, it's an acetylamide. Acetamide group here, the acetylamide group here is removed, and you put this acetyl group, you replace it with a hydrogen. That is what you call aniline. So if you get aniline, you remove that group here, you cleave there, and you put a hydroxyl group there. That drug is still, or you put a hydroxyl group there to get the parahydroxy. That drug was still found to be highly toxic. And of course, if a drug is highly toxic, it has no use. It has no therapeutic indication. Then esterification. This is esterification because you are making an ether, you are having a hydroxyl group, and you are adding a methyl group or an alkyl group. Esterification of the phenolic function with a methyl or propyl groups produces derivatives with greater side effects than ethyl groups. And that is why you have phenacetin. So this is just explaining why phenacetin, why the choice of an ethyl group and not a methyl and not a propyl, because both methyl and propyl produced highly toxic drugs, but ethyl which gives us phenacetin, was much safer. Substituents of the nitrogen atom that reduce basicity, reduce activity, unless the substituent is metabolically labile, such as acetyl. All we are saying is that, again, if you esterify, if you actually acetylate, because that's what has been happening. If you look at the amino group and you acetylate that amino group with groups that cannot be cleaved, again, that led to inactive compounds. Like amides derived from aromatic acids such as N-phenylbenzamide are less active or inactive. What we are saying is that once the group that you put at this position, even for paracetamol, you cannot put any group here. Like if you decide to put a C double bond O and a phenyl, if you replace the methyl group with a phenyl ring to give you the phenacamide, that drug was found to be inactive. So the only drug which was found to have good activity is where the amino group of para-aminophenol was acetylated, or actually was derivatized through addition of an acetyl group, or causes addition of, this must have been acetic acid, derivatized by formation of the acetanilide. That was the only group that was found to give us activity. Putting two large groups or groups that cannot be hydrolyzed, that resulted in no activity at all. And that is why also paracetamol, as we are going to see, because of the fact that it can't be hydrolyzed in the body, to release to the aminophenol or even to be oxidized, it can actually form also very toxic metabolites. And we are going to discover that also paracetamol, though we take it for granted, is a very safe drug. You must be careful because it can also cause, it can kill, it can cause serious toxicity. And we are going to look at that in a minute. It is weakly acidic, of course, that's because of the phenol and synthesized by acetylation of para-aminophenol. It is weakly bound to plasma protein, indicated for use of antipyretic analgesic activity. It does not possess anti-inflammatory activity. It's available in various formulations, which is very good. You can have it in suppositories, you can have it in tablets, capsules, granules, and even solution. Paracetamol solution, like cowpaw suspension, and of course, very, very good for children, especially if they have fever or pain. Remember, more often than when you have fever, more often than not, you also have inflammation, and therefore it will take charge of the pain because of the inflammation. And of course, the doses that much of that you will do in pharmacology, but we are saying doses of greater than 2.6 grams per day are not recommended for long-term therapy because of potential hepatotoxicity. So this is one of the areas. So paracetamol, a drug that you think is very safe, and you can take as many as you wish per day. We are only learning today, and it's important for us because even when you advise clients, we are only learning that it can be highly toxic. And in a minute, we learn how paracetamol can cause its toxicity. Unlike aspirin, is stable in aqueous solution, making liquid formulation readily available, particular advantage in pediatric cases. Of course, we do have paracetamol syrup. Remember, syrup and suspension are different. We do have paracetamol syrup where, of course, the paracetamol is in aqueous solution. We do have, of course, for cowpaw, it's a suspension. And we are saying aspirin, it can be a challenge to make aspirin syrup because aspirin, being an ester, look at this, paracetamol is an amide. Amides are stabilized by resonance, as we have discussed. So they are more stable in aqueous media than esters. An ester highly degrade. It's highly hydrolyzed in aqueous media. Now, from that, now I have said not much in this group, because the only important group in that drug here is paracetamol. And therefore, let us now look at metabolism of paracetamol, because actually the toxicity of paracetamol originates from the root of metabolism. And you find undergoes glucuronic conjugation, 85%. You already, when you look at paracetamol, it's already a group, and therefore readily undergoes glucuronic conjugation. And that once it is conjugated, it is easily removed from it. It can also undergo sulfate conjugation. So paracetamol can undergo glucuronic conjugation or sulfate conjugation. But as we discussed earlier, more often than not, glucuronic conjugation predominates. I did mention that the enzymes that catalyze glucuronic conjugation are non-selective. There are many. They are non-selective for various substrates. So you can find there are many and they are non-selective. We also did mention that the precursor of the glucuronic acid, which is being conjugated with the substrate or the drug, is readily available because it comes from glucose. So the ready availability of both the enzymes and the moiety that is required, or the activated moiety which is required for conjugation, glucuronic conjugation, makes it readily available, makes it more popular than the sulfate conjugation. The sulfate conjugation, the enzymes that are responsible for sulfate conjugation, and even the compounds or the activated moieties that you require for sulfate conjugation are a little bit rare, and therefore you'll have more of glucuronic conjugation than sulfate conjugation. Number two, in children, you find that the glucuronic conjugation in children, very neonates, we call them neonates, the very young ones, the neonates, you find that they normally metabolize most of the drugs through sulfate conjugation because the complex, the glucuronic conjugation system is quite complex and it requires time to be fully developed. So you find that they normally metabolize through sulfate conjugation rather than glucuronic conjugation until the metabolic pathway for glucuronic conjugation, that's the enzymes that are required for glucuronic conjugation, are well developed and the glucuronic conjugation can take place. So you find that 85% of this drug is metabolized by conjugation. But in rare occasions, you can that this drug can also be metabolized through oxidation by cytochrome P450 enzymes, and specifically cytochrome P450E1 that can metabolize the paracetamol when it takes this alternative route, because the main route of metabolism, as you can see, is conjugation. When the metabolism of paracetamol through cytochrome P450, the metabolite N-acetyl para-benzoquinone imine, this should be imine, and I here benzoquinone imine, N-acetyl para-benzoquinone imine is highly toxic. This metabolite is highly toxic. It is a very strong electrophile, very strong electrophile, which means it is electron deficient, and therefore it looks within the body which have high density of electrons. And in the body, especially where you have oxygen, you have sulfur, those are sites of high nitrogen, those are sites of high electron density. So you find if this highly toxic electrophile is not removed from the body, then you will find it can bind the tissues within the body and give you toxicity. In where it's a little bit of it, where the concentration of this is within manageable levels. In other words, you've taken paracetamol and a bit of it is formed into this toxic metabolite, you do have what you call glutathione. And I believe when you are doing metabolism, if you have done metabolism, I hope you are told that also glutathione is used within the body as an antioxidant. It is used in the body to reduce toxicity or to mop up toxic electrophiles. One of the roles of glutathione is actually to reduce this compound. Glutathione in the reduced form will help in the reduction of this compound. And once this compound is reduced, the highly oxidized compound, once it's reduced by glutathione, then it ceases to be toxic and it is readily removed from the body. So what we are saying is that the toxicity of the paracetamol is metabolism, and we will see what are the risks, what factors increase that risk of getting the toxic metabolite, or what factors decrease that risk. So we are saying normally 85% metabolism by conjugation. There is a minor route of oxidative pathway by cytochrome P450 enzyme, especially the 2E1 and 3A4. So if it is metabolized through these enzymes, then you will produce toxic metabolite N-acetylparabenzoquinone imine, very highly toxic electrophile, which if it is going to be removed from the body, it requires detoxification by use of glutathione. So if, because of other reasons, you have low glutathione levels, or you have exhausted your glutathione levels,